Golgi complex. See, the Golgi complex is actually a cell organelle, and it is present mostly in eukaryotic cells. In 1897, it was discovered by Camillo Golgi, and that's why the name of this organelle was given as a Golgi apparatus or a Golgi complex or a Golgi bodies. Now, see, if we see the structure of a Golgi complex, you will find that the structure is it is a membrane-bound organelle and made up of three types of structural features: cisternae, vesicles, and tubules. Now, we will see one by one the cisternae. The Latin meaning of cisternae is a collecting vessel, and this is a saucer-shaped flattened membrane, includes sacs. They are arranged one above the other, forming a kind of a stack, uh, and these stacks are independent, they are not interconnected. Now, when we talk about the number, you will find that very few number of cisternae will be present in a plant cell, but in case of animal cells, 10 to 15 number of cisternae that may be found in a plant cell. Then, if we consider the shape of the Golgi complex, you will find that the Golgi complex has a peculiar type of a shape. It is convex at the one end and concave at the another end. Now, this concave and convex ends, they are oriented in a typical manner. The convex side of the Golgi complex is towards the endoplasmic reticulum or nucleus, while the concave type of the region that is present towards the plasma membrane. Now, another peculiar feature of this is that this convex type of um, region that is also called as a forming phase or a cease phase. Now, why it is called as a forming phase? Forming phase is because the whatever the vesicles that are formed on a rough endoplasmic reticulum, these vesicles are um, uh, or they aggregate at the forming phase, and they aggregate, and after some time they fuse with each other. So when they fuse with each other, they form a larger vesicles, and then these larger vesicles then again fuse with each other to form a cisternae or a stack, one of the stack of this Golgi complex. So that's why this particular region is called as a forming phase or a cease phase. Now, as we move from the cease phase to trans phase, now here you will find that the stacks, or we can say the cisternae, they give rise to the smaller vesicles, and these vesicles are they bud out of this cisternae, and that's why this particular region is called as the maturing phase or a trans phase. Now, why it is called as a maturing phase? That is because that whatever the vesicles, now these vesicles contain, uh, uh, we can say that the whatever kind of secretory material that is present in these vesicles, and these vesicles they come out of this particular cistern. That's why it is called as a mature phase or a trans phase. Now these vesicles they fuse with the plasma membrane, and they pour their content outside. So like that, you will find the shape of peculiar shape of this Golgi complex. Then, if we see the content of this, The inside a cisternae or a cisternae contains a kind of a fluid, and that fluid is called as a matrix. Now this fluid or matrix contains very different kinds of molecules like proteins, lipids and carbohydrates. Now out of these proteins, lipids and carbohydrates, the proteins are very important, and there are two types of proteins are present in this Golgi complex. One is called as a residential proteins and another is called as a cargo protein. Now, why such a kind of a name is given to the proteins? That is because, see, proteins which are resident of that particular um, Golgi complex, the such type of proteins they are called as a residential proteins, and those they are in the travel. That means they are synthesized in a endoplasmic reticulum. Then they travel via vesicles to the cis phase of the Golgi cisternae or a Golgi complex, and then they form a cisternae. Then this cisternae then move towards the trans phase, and from trans phase this cisternae leg out, or we can say they give rise to a smaller vesicles, and these vesicles then fuses with the plasma membrane. So uh, this what is the purpose of this? This purpose of this is that whatever the proteins that are synthesized in endoplasmic reticulum, they are transported to the outside of the cell or up to the plasma membrane, and these proteins which are incorporated in a plasma membrane, or these proteins which are secreted outside the cell, these are called as a cargo proteins, because they move from the endoplasmic reticulum to the plasma membrane via this matrix, or sorry, via this Golgi complex, and that's why they are called as a cargo proteins. So this is the reason. Then another type of structural feature is the vesicles. It is a membrane-bound spherical structures, and they bud out, or we can say that the phenomenon of budding out of these vesicles from the Golgi system is called as a blebbing. So they come out due to the blebbing, and these vesicles they form or they transport proteins, as I have already said to you, that the cargo proteins which are there, these cargo proteins are then taken to the vesicles, and from vesicles they are taken to the plasma membrane. So these vesicles are there, they are spherical structures. Now when we talk about the third typical structure of a Golgi complex, that is the tubules. So in some cases, the tubules are found on the cis and trans side of the Golgi complex. So if they are present on the cis side, then it is called as a cis Golgi network. If they are present on the trans side, then it is called as a trans Golgi network. So these tubules are there. So this is how the structure of you can say the Golgi complex is there. You can see here in the diagram also. This particular region is called as a cis Golgi network. It is a forming phase. Uh, this particular region is called as a trans Golgi network (TGN), and it is a maturing phase. So, like that, these are the um, we can say the stack cisternae. Now, another peculiar feature of this is that this cisternae they are also divided into three types of cisternae. The cisternae which is present on the cis side, it is called as a cis cisternae. The cisternae which is present in the middle region of the Golgi complex, it is called as a middle cisternae, and the cisternae which is present on the trans side of the Golgi complex, it is called as a trans cisternae. So this is how that um, the, there is a kind of a difference within the um, Golgi cisternae also. So this is a overall structure of Golgi complex. As I have told you that the Golgi complex it has a specific orientation. The Convex side of the Golgi complex is towards the endoplasmic reticulum, and this convex side is also called as a forming phase or a cis phase. The cis phase contains the vesicles, and these vesicles are coming out from the rough endoplasmic reticulum. These vesicles they coalesce, fuse with each other to form a smaller or larger vesicles, and these larger vesicles then again fuse to form larger vesicles. These larger vesicles fuse to form the cisternae. So, like that, these are the cisternae which are present on the cis phase. These are called as the cis cisternae. These are called as the middle cisternae, and the cisternae which are present on the extreme end of the Golgi complex or towards the plasma membrane. They are called as the trans, or we can say trans cisternae. And this particular phase is called as a maturing phase or a trans phase. Now, uh, the tubules and vesicles that are present on the cis side, they form a kind of a network that is called as a cis Golgi network. Tubules and vesicles that forms a network-like structures on the trans side also, trans phase also. These are called as a trans Golgi network. So these are the vesicles that coming out of the um, Golgi cisternae, and this is all about the structure of a Golgi complex in all. So, of a Golgi complex. Now, another two special features or two structural features of a Golgi complex that is also seen. One is the orientation, and another is the dynamic nature of the Golgi complex. Now, here we have already discussed about the orientation. See here that the Golgi complex has a typical type of orientation. You will find that the convex side of the Golgi complex is towards the nucleus, and the concave side of the Golgi complex is towards the plasma membrane. So such a type of orientation of this is seen in a Golgi complex only. No other organelle, cellular organelle, shows such kind of a orientation. Then another typical feature is the dynamic nature. You can see here each cisternae is different from the other one, and another peculiar feature is that this cisternae after some time it will reach here. This cisternae after some time will reach here, and this cisternae after some time will reach here. This cisternae will transform into the vesicle. So you can imagine that the vesicles which are synthesized or they bleb out from the endoplasmic reticulum, these vesicles they coalesce to form this cisternae. Like that, this cisternae again move to forward to towards the plasma membrane side. That means towards this side, towards the trans side, and you will find after some time this cisternae will be there. This cisternae will be here. Like that. That means the Golgi complex does not have a static structure. It has an ever-changing structure, and that's why it is called as a, or it has a dynamic nature, and that is the peculiar feature of the Golgi complex. So these are the two structural features of the Golgi complex. Then, if we see the functions of a Golgi complex, the Golgi complex is considered as, or it is nicknamed as the what we call it as the traffic police of a cell. Now, why it is called as a traffic police of a cell? That is because whatever the proteins that are synthesized in a smooth endoplasmic reticulum, lipids synthesized in a smooth endoplasmic reticulum and proteins synthesized in a rough endoplasmic reticulum, these proteins or the lipids, they are transported to the various parts of the cell, even to the outside of the cell, and that is through this Golgi complex. So the processing, packaging, and transportation of this particular synthesized material or a product, that is towards its particular destination, that is done with the help of a Golgi complex, and that's why it is called as a um, sorting organ, or we can say the traffic police of a cell, because we know that what is the function of a traffic police? To govern the traffic on the road. So similarly, here the whatever the proteins that are synthesized, the distribution of these proteins that is totally with the help of a Golgi complex, and that's why Golgi complex is considered as a traffic police of the cell. Now, first function of this is protein processing and modification. As we know that whatever the proteins are different kinds of enzymes that are synthesized in a endoplasmic reticulum. Even in case of a smooth endoplasmic reticulum also smooth endoplasmic reticulum takes part in synthesis of lipids. So the proteins and lipids that are synthesized in endoplasmic reticulum that are transported to the Golgi complex. Here there is a modification of certain kinds of proteins and lipids that takes place. Now, this modification or the processing, that is by addition or removal of various kinds of carbohydrate molecules. Now, that may be the monosaccharides. So, mostly monosaccharides are of different types. So, the addition of these different kinds of monosaccharides on lipids and proteins, that is done in a Golgi complex. Uh, so the second function of Golgi complex is a lipid modification. The endoplasmic reticulum synthesizes a lipid that is known as a ceramide, and with this ceramide is transported to the Golgi complex, where certain kinds of enzymes, these are known as glycosyl transferases. These enzymes are responsible for putting or adding a glucose molecule to the ceramide. Such kind of ceramide is called as a glucosyl ceramide. This glucosyl ceramide is transported to the plasma membrane, where it is incorporated in a plasma membrane. Now, this glucosyl ceramide is responsible for the formation of cell surface markers, and these cell surface markers are very important in cell recognition. So this is how that different kinds of lipid molecules are modified with the help of enzymes that are present in the Golgi complex. Then, as we know that there are certain kinds of proteins. These proteins are multi-subunit. That means number of subunits are there in a particular kind of protein. For example, in case of insulin, the number of subunits are there, and these subunits are joined together by the sulfide bond. So, addition of a sulfur, that is with the help of this Golgi complex. So what that process is called as the sulfonation, and in the Golgi complex, certain kinds of enzymes that are responsible for transferring sulfur molecule to the protein chain. So the in a protein we know that proteins are made up of amino acids, and one of the amino acid is the cysteine. So the two subunits are joined by the disulfur bonds, and this is how that the sulfur is added to the protein. So that is, or this kind of a modification is done in a Golgi complex. Now, the fourth one is the phosphorylation of a protein. So the proteins which are to be transported to lysosomes are called as the lysosomal proteins, and these lysosomal proteins are phosphorylated before transportation to the lysosomes. In a Golgi complex, these proteins are phosphorylated, and then they are transported to the we can say the lysosomes. Now, these lysosomal proteins. Now, how the identification is done? When they are phosphorylated, the three-dimensional structure of this lysosomal protein that takes place, and because of that, we can say a three-dimensional structure is identified by the certain kinds of receptors, and these receptors, or we can say along with these receptors, these phosphorylated proteins are then transported to the lysosomes. So like that, the phosphorylation of a protein that is also done in a Golgi complex. Then synthesis of a cell wall polysaccharides. We know that in case of plant cells, uh, the hemicellulose and pectin are synthesized in a Golgi complex, and these hemicellulose and say pectin, it is transported to outside of the plasma membrane with the help of the Golgi complex. That means this is how whatever the polysaccharides and pectins that are synthesized in a Golgi complex with the help of vesicles, they are transported to the outside of the cell. So this is a synthesis of a cell wall polysaccharides. Then sixth function is lysosome formation. I have already told you that the Golgi complex is responsible for modification of certain kinds of proteins, and these modifications, uh, these modifications are responsible for taking up that particular protein to a particular destination. So here we will see that. See what happens in a endoplasmic reticulum. You will see that the step first is a glycosylation of this particular lysosomal enzyme that takes place. So what do you mean by glycosylation? Addition of a sugar molecules that takes place in a endoplasmic reticulum. Now when this lysosomal enzymes then are taken to the cis Golgi, here, what happens that the second step is the phosphorylation of a lysosomal enzyme that takes place. So here a phosphate molecule is added to the uh, lysosomal enzymes. Then these lysosomal enzymes, along with this phosphate molecule, they are transported to the trans Golgi. Now, in a trans Golgi, another kind of a enzyme is present. That enzyme is, or we can say, a receptor is present, and this receptor is responsible for binding with the phosphate molecule. Now, after binding, or this receptor is specialized for binding with the lysosomal enzymes which are attached to the phosphate. So, this receptor then forms a vesicle that this fourth step is a vesicle containing exclusively receptor bound lysosomal enzymes that bleb out from the uh, transistor. Now, after that, the next step is once the lysosomal enzymes are segregated, then the receptor go back to the trans Golgi and is recycled to bring another carbon. So here and again, this particular receptor is separated from the main vesicle, and this is how that this particular lysosomal enzyme is segregated in this particular vesicle, and now this is called as a lysosome. So this is how there is a mechanism of sorting out of lysosomal enzymes in a lysosomal vesicles. Then the last one is the formation of acrosome in the sperm. Golgi complex is also responsible for formation of acrosome in the sperm. Whatever the lytic enzymes that are synthesized in a Golgi complex, in endoplasmic reticulum, they are modified in a Golgi complex, and then these particular vesicles that comes out of the Golgi complex, they form they coalesce with each other to form a larger vesicle that is called as acrosome, and this acrosome is responsible for accumulation of hydrolytic enzymes, and these hydrolytic enzymes are used by the sperm for fusion with the female gamete. So, like that, the formation of acrosome that is done with the help of a Golgi complex. So, this is all about the Golgi complex. If you like my video, please subscribe to my YouTube channel, BAMU'S Biogenius. Thank you. Thank you.